Hello, everyone, and welcome. We are just getting started with the Business of Alt Protein monthly seminar. My name is Lane Clark, and I'm the Innovation and Entrepreneurship Manager for the Good Food Institute. Today, we'll be diving into the development and scale-up process for precision fermentation. We'll be touching on everything from strain selection to downstream processing and covering the do's and don'ts at each stage. Before we begin, I have a few housekeeping items to go over quickly. First, for those who are unfamiliar, the Good Food Institute is an international nonprofit organization developing the roadmap for a sustainable, secure, and just protein supply. We identify the most effective solutions, mobilize resources and talent, and empower partners across the food system to make alternative proteins accessible, affordable, and delicious. Second, the seminar will be recorded and posted to our YouTube channel. Additionally, a copy of the recording will be emailed to registrants after the presentation. You can also view previous seminars on our YouTube channel. And third, the seminar will include an audience Q&A for the last part of the hour. Please ask your questions in the Q&A box rather than in the chat. You're welcome to ask questions throughout the seminar, but we will address them towards the end. And finally, immediately following the seminar, we will be hosting a virtual networking session via the Meetaway platform. We'll drop the link in the chat that will take you to the registration page where you can sign up if you haven't already. This is a great opportunity to meet other professionals in the alt protein industry. You'll be matched with several other people for one-to-one conversations. I have met some fantastic people at these mixers and encourage everyone to join us. Just remember, you need to sign up beforehand. Okay, so let's get started in earnest by introducing our wonderful panel. And I am losing my notes just a second, sorry. Sorry about this. Okay, so today's seminar will feature the team at Center for Process Innovation. CPI is an independent deep tech innovation center located in the UK. Founded in 2004, they're a founding member of the UK High Value Manufacturing Catapult. Their team of 700 plus people work with companies to accelerate the development and commercialization of innovative products. Today, we have 3 team members from CPI, Kang Lee, Jonathan Kennedy, and Stephen Wright, who will dig into the scale-up process for precision fermentation. I'll now hand it over to the CPI team to take it from here.
Okay, thanks, Len, and thanks to everybody from GFI, Audrey and Molly, for inviting us to give this presentation, and thanks to everybody who's attending. So I'm just going to now share my screen. Okay, so what we're going to be talking about today is as Lane told us, is navigating the upstream to downstream process in precision fermentation. I'm Jonathan Kennedy. I'm the Holster and Development Manager at CPI, CPI's biotechnology unit. And my background is in molecular biology and strain engineering. Chang and Steve will give a brief introduction to themselves when they come to their part of the talk, but I'm just going to give this starting introduction to what CPI, a little bit more about what CPI is, and then tell you a little bit about my role in CPI and how it relates to precision fermentation. Okay, so where CPI sits is really what our focus is in this innovation space between research and invention and the commercial market. And what our objective is to help businesses take their bright ideas and turn them into commercial reality. And so we do this by using sort of our physical assets, we have a wide range of different equipment and facilities across our different business units, and probably more importantly, with our expertise in using those assets to really commercialize and scale up processes that our customers come us with. We've worked on a wide range of things, and essentially, it's this element and the experience we have with working with a diverse range of companies coming across a lot of problems and understanding really what's needed and to turn those into sort of commercial realities and coming up with solutions to that. So as Len, we're located in the UK. mainly most of our facilities are in the northeast of the UK, and that's where me, Stephen, and Chan are currently sitting in our site at Wilton in our biotechnology business unit. We also have a number of other facilities in the northeast of England, with also some additional facilities in Scotland and some other offices throughout the UK. So at these different facilities, where we are, as I mentioned before, we're in the biotechnology business unit. We also have in our other facilities focus on different technologies such as biologics, on formulations, on electronics, and on medicines manufacturing. So we really look at a wide range of technologies to address the sort of markets and businesses where we can help them in different areas. So what we're talking about today is our food, feed, and drink offering, our focus on there. What we see, so we've seen at CPI that the food and feed market is really somewhere where there's an awful lot of innovation in the area and a lot of opportunities. And yeah, and it's really what we want to look at is to see where CPI can help companies and really help turn these really good opportunities and into commercial offerings. So in our biotechnology business unit, we're really focused on the actual producing the products there. So looking at the alternative proteins or functional ingredients. And here we've been investing in our facilities here. We have some lab scale food grade laboratories for making samples at lab scale, but we're also heading towards accreditation for pilot scale production of food grade products. So, and to go hand in hand there, it's also all about sort of training our staff as well. So we have a team of HACCP trained staff for working on these food and feed projects. Across our other business units, we also have complementary expertise in terms of sort of formulation that can be used for the food and feed markets, and also across logistics and packaging. So the idea is that it's not just producing the materials, but it's also we can help with the formulations and later on in those development cycles as well. So a little bit more about CPI. Lane gave a bit of an introduction there. So we've been around for over 19 years. We've been growing all this time. According to here, we've got 600 staff. I think Lane said 700, so maybe we've grown a bit since this slide was made. I don't know. And we've got about 1,300 completed projects through that time, and our annual turnover is growing every year. So that's kind of the end of the sales pitch for CPI. Yeah, so now I'll move on to what we do in terms of the science. So my role at CPI is in the whole strain development group, and so what I'm going to talk about here is where that whole strain development fits into that precision fermentation market. But for what I want to just start off with initially is really what do we mean by precision fermentation? So you can see from this slide here that what we're really looking at is producing the same kinds of products, traditional food products that are produced by traditional farming. So here's the examples are essentially sort of dairy products, and what that are produced by having a traditional farm and here's your traditional harvesting process, and then this is processed into the milk, cheese, and butter and things that we all love and know. So the idea behind precision fermentation is that really what we want to do is produce those very same products, but by using fermentation technology. So instead of starting with cows growing in a field, what we have are bacteria or yeast or filamentous fungi in which we've inserted genes that encode for those same milk proteins or whatever proteins you're interested in. So they were producing essentially the same protein. Instead of the cow being your fermenter, what we have is stainless steel fermenters to produce those, and then through the magic of downstream processing, turn those into these sort of same food products. So Yeah, as I mentioned, the precision fermentation is really all about the production of specific food products and ingredients using microbial fermentation. It almost always would involve a synthetic biology step to generate a microorganism that's actually programmed to produce that final product. The other important point about this process is that it's really a well-established, the technology underlying precision fermentation is really well-established for the production of biochemicals, enzymes, and pharmaceutical products. But what we're doing here is just using that same technology to produce the food products. And the time now it seems to be ready for producing these food products by these means. There's a lot of environmental and social reasons for that. There's as I'm sure we're all aware, there's a lot of pressure on the food production system from population increases, climate change, and what precision fermentation, the opportunity here, provides us a way of producing nutritious food that's non-seasonal, using minimal land and water use, without the use of herbicides, pesticides, or fertilizers. And so essentially these products could theoretically be produced anywhere in the world at any time of the year. In terms of it being the right time, there's certainly a greater acceptance throughout the world of alternative sources of food, and there's also a greater emphasis on animal welfare, and there's increasing people drawn to sort of veganism or flexitarian sort of diets. In terms of the products that can be made, I think virtually any naturally occurring product can be produced in this way. We're focusing this talk more around the proteins, such as the milk proteins like lactoglobulin, or egg proteins like ovalbumin. But there's also a wide range of other products that can be produced by food products that can be produced by precision fermentation, so enzymes. So chymosin, this is probably I think it's the first, the first food product that was produced by genetic engineering, and this is widely used in the production of cheese, and this has been around since the 1990s. There's also products such as sweeteners, protein sweeteners or other sweeteners, and then other small molecules that flavorings or pigments, and also the fats and oils, which are another important product that can be produced by precision fermentation. So where do you start in this process? Well, from my perspective, where you start is considering what you are going to use as your host strain for this. And probably the most important aspect of that is the safety and regulatory aspects. So having a microorganism that is recognized as safe, that's got a history of safe use, and that would also have some sort of consumer acceptance is probably the most important starting point for this. The second most important thing to consider is really, what is your target product? So is the target product an ingredient, or is it the whole thing, the final product? Is it a protein? Is it a lipid? Is it another metabolite? What's the market size? And then also thinking about, what level of purity you need in that product as well. And I think those two things are probably the most important things to consider when you're thinking how you're actually going to produce it. But there's many other things we need to consider as well as part of this. And this is really why, why we've got everybody involved in this, the whole team at CPI, is we've got to think about the feedstock. The feedstock is a big element of the final cost, but it can also have an impact on the actual product itself. We've got to think about the scalability of the process, and Chan will be talking more about that. But essentially, you've got to think about how that microbe scales and what's the history of that. We've also got to think about the downstream processing that Steve's going to be talking about as well. And from my perspective, we've got to think about the genetic amenability of the strain. So what are the tools available for that strain? What's its stability? And then other aspects of that would be, whether it results in a glycosylated product, whether there's particular problems with proteolysis. So there's a lot of things to consider. And I think from putting all these things together, you can come up with some ideas of what you can use and take forward. So in terms of that strain development process, having picked a strain or strains, what we wanted to look at next is really how do we get that strain to produce the product that we're interested in. So this is eventually, this obviously involves expressing those proteins in that strain, and for that you can use either plasmid or chromosomal systems, and those have pluses and minuses. The chromosomal system would obviously give you a very stable expression system. Looking at things from a plasmid point of view, it's a little bit quicker. It can produce in higher yields, but it can be less stable. In terms of the expression system that you're going to use, first thing to consider is the promoter, whether that's an induced or a constitutive promoter, and what promoter you use then really has a big impact on the fermentation process that you're going to use. So this is why you've got to think about all these things at the start. Other things to consider are the secretion of the protein. So that would be another thing to look at. And the other important factor for food grade systems is really you want to have an antibiotic-free system. In terms of looking at all those options, obviously this gives you an awful lot of things that you can consider. And in order to really find what is the best for your purpose, it's important to have a very, very robust screening process. And so for this, what we would recommend is certainly initially doing some very small scale screening to narrow down your process, initially using simple sort of microwell plates, but then also as soon as possible, we would recommend going through to more fermentation-based systems, such as microbioreactors, such as the Amber 15 or Amber 250 systems, that would allow you to assess strains at fairly high throughput in more process-relevant conditions, because essentially what you're going to be producing isn't going to be the equivalent of a microwell plate or a shake flask. In terms of that analytics, initially, really what you're looking at is the protein that you're producing. You'd be interested in the yield, but also in the integrity of your product. Is it what you're expecting to produce? Is there any problems with proteolysis? Is glycolysis a problem? So those are other aspects that would come into that selection process. And it's very likely that in this process, you're going to have to do this multiple times. It's going to be an iterative process. And after a few rounds, hopefully you get to something that really is starting to look like it could be a strain to take forward to the next stage. And that's where I'm going to hand over to Chan.
Yeah, thank you very much, Johnson. Good evening, good afternoon, and good morning, depending where you are in the world. My name is Chang. Very pleased to be able to speak about the upstream process development work that we have done in CPI and what we can do to help the client to scale up and commercialize their process. I'm principal scientist, joined CPI for 6 years. My background is about chemical engineering. Maybe I'll just start my presentation by talking about what traditional process development looks like. Typically, they use a non-integrated approach. It looks quite linear, starting with proof of concept, developing strain, without thinking about the scale-up issues. The challenge for that typically falls over when it comes to scale up because of the extensive metabolic stress that cells experience at scale up. And the consequence of that, they may have to accept the suboptimal process and move to the production. As a result, the product title and the yield may suffer. This might be okay for the industry like a pharma or cosmetic, where the margin of the profit is high, but definitely a no-go for food and feed, where the yield and productivity is everything. So that the process may not be as linear as it's supposed to be. Quite often we see people coming back to the development stage and they have to try different strains, different process options, just in order to overcome the commercial barrier they're facing at a larger scale. And as a result, you could imagine that the overall time take to reach the market can be as long as 12 to 15 years. and a lot of investment needed to get to the market. So I just mentioned in the previous slide, metabolic stress. So what is metabolic stress and why is it? So you could imagine in the larger scale fermenter, the substrate like glucose and pH tetron added from the top of the fermenter, where the compressed air, which also acts as a substrate to aerobic fermentation, is added from the base of the fermenter and rising to the top before it's leaving the fermenter from the exhaust lines. So as one could imagine, due to the specific ways of adding substrates to the fermenter and the size of the fermenter, the concentration gradient do exist in large fermenter. These include several critical process parameters, for example, glucose, dissolved oxygen, pH, dissolved carbon dioxide. So their concentration gradient in that fermenter will impose significant environmental stress to the cells. As a response, the cells may behave very differently in the lab, where the homogeneous mixing is prevalent and no such stress can be experienced. As a result, the process, what we say that over-optimized in the lab, sometimes may fail over at larger scale. And this is the typical things that people fail to consider when they move their process to commercializations. So in contrast, we do things slightly different. We would like to do the scale up using the integrating the solutions and like to try these solutions as early as possible in the process development journey. So the typical things and tools that we have developed in the past, these include wholesale strain and screening. Jonathan has talked about this at length. We also do design experiments, rational scale down, computational fluid dynamics, and collectively, we would say that by considering the risk at the scale up, the journey to commercialization can be greatly shortened. We have seen successful examples where it only takes as short as 1 to 3 years to reach to the demonstration scale from just proof of concept on the paper. Today, due to the limitation on the time, I was just talking about the rational scale down and the computational fluid dynamics. So these slides is all about the fermentation scale down. In a nutshell, what fermentation scale down focus is to identify a suitable small scale fermenter and its associated running conditions, which allow us to predict and mimic the larger scale operations so that we can generate the same quality of materials or process data, which help us to navigate through the scale up process. So typical things, people would say that the mixing and mass transfer are the most prominent process parameters to consider with scale up. And sometimes not very difficult it's not very easy to achieve the same scale up effect in both process parameters. Sometimes a trade-off needs to be made. Another thing you need to consider is the gas sparging. In a larger scale reactor, typically the air bubbles are bigger and less uniform than the small scale. Heat transfer is less issue in the large scale fermenter where it's the heating cooling coils being used. But we could imagine that if this strain is particular mesophilic, that is to say they grow at ambient or slightly above ambient temperature, the heat transfer requirement may be high, and there are practical consideration on that side as well. Generally speaking, we have tested our small scale reactors and identified a 5-liter fermenter, which gives good enough mimicking and predicting power with large scale fermenter. And we have tried E. coli based process and demonstrate that process can be directly scaled up from the 5-liter to 750 to 10,000 liters without any significant technical barrier. And since we have used the scale down models to help a number of clients to scale up their process successfully. The next thing I'd like to talk about is the computational fluid dynamics. So we have applied commercially available CFD software and fully characterized all of the stir tank that we own in CPI, able to simulate a number of process parameters we deemed relevant for the batch processing. These include the liquid mixing, mass transfer, gas hold up, bubble breakup, the coalescence, power input, turbulence, shear, velocity profiles, etc., etc. So the benefits doing CFT is allow you to do in silico experiments to understand the process parameters. That is usually hard to measure. And also more importantly, it also gives you a profile of these parameters. So that allows you to understand the temporal and spatial variation of these process parameters. Sometimes we find it's more critical when you're running large fermenter than just some gross measurements. So these things are hard to achieve with analytical suite that typically associated with fermenter, but can be rapidly achieved by using CFD modeling. The next slide, I'd just like to talk about briefly about continuous fermentations. As we know, continuous fermentations are the predominant form for biomass alternative protein fermentations. We have examples of commercial productions like corn process, but it hasn't been used successfully in the preceding fermentations for practical reasons. Although it's not the case yet, we use continuous fermentation extensively in our laboratory for development purpose because we found it very useful tool to assess the stabilities of genetics, media, and product, and also to identify the root of contaminations and come up with mitigating plans to reduce the risk of contaminations. We also use that to evaluate genetic modifications. We did a lot of chemostat studies to understand the impact of genetic modification on the cell physiology. We also use these techniques to assess the bioprocess control strategies to test some of the new latest bioprocess technologies, which has the potential to greatly improve the productivity. We also use this to test the process models. Last but not least, this emerging technology in artificial intelligence, and we are trying to incorporate this AI using the continuous fermentation and associated integrated biosense, integrated automation and biosensors. Okay, so I hope that gave you a great flavor of what it looks like for modern and integrated upstream process development. Now, happy to pass on to my colleague, Steve, to talk about specifically about the DSP challenges.
Hello, everyone. Yeah, just a quick introduction, because I know time is tight here. So my name's Steve Wright. I head the downstream processing team here at the Biotechnology Business Unit for CPI. My background is I'm a biochemical engineer, and I've been around the pharma and the industrial biotech industry for around 20 years. So what I'll do is I'll go through some of the issues that I've come across on projects, talk about the landscape a little bit, and some of the challenges that I tend to find in the downstream processing of a lot of these precision fermentation products. So this first slide goes through similar themes that Jonathan and Chang have talked about, but it's something that I've kind of noticed a lot in recent years, and the shift that we need to do. So typically you tend to have the upstream development path set. So that's from, cloning the genes, getting the vector system right. That would be Jonathan. You would then go through process development in terms of the screening, and then it would basically be hit over a fence over to DSP. And what I'm trying to get at here is that it's now meant to be a more integrated approach, where the actual starting from the gene of interest into the vector system should actually be with all disciplines, not just the upstream and the molecular team, because when you do the downstream processing, there are lots of improvements that can only be made really at the gene scale. So having inputs really early on makes downstream processing so much easier. So kind of an example is if you express any of your biomolecules, if it's secreted into the surrounding media, it makes downstream processing so much easier. So doing that early on, getting the right titers at the end of the fermentation means that there's more leeway in the downstream processing. So it's more of an integrated approach. So that was the first message from this. The second message is actually knowing where you want to finish, which is kind of what we try and talk to a lot of our clients about. So if you know where you need to be, if you need to be at a certain amount of material that you want to generate, to be a commercially viable process, if you know what kind of price point you want to be at, that allows the whole process to be tailored towards that endpoint. We tend to find a lot of the startups, the SMEs come, they have a really good idea, they have the proof of concept, but when you look at it from a scale-up perspective, to get where they want to get to, it becomes unmanageable and it costs actually more to produce than just to sell it. So if you go right, and it costs more money to start again. So if you can get that integrated at the start, know where you want to finish up, you can then integrate that right at the start of your development process. So when I think about precision fermentation, like Jonathan's touched on, it's been around for quite some time, 40, 50 years. So the first commercial product was in the 80s for insulin. But the first thing you notice when you talk about that is it's for biopharmaceutical. And they are really high value products. And then if you talk about precision fermentation for food, which is essentially it's the same technology, but you're also using the same technologies for the downstream processing. So the first question is, how do I produce a product that's low value and maybe mid value, using the same technologies that initially used high value products? And how does that become cost effective? So that's the first hurdle you have to overcome. The second one to think about also is that with these existing technologies been around for the last 30, 40, 50 years, is that when you talk about biopharmaceuticals, you're talking about small quantities if it's injectables, whereas for food, you're talking grams, kilograms, tons of material, which is completely different when you're talking compared to therapeutics. Again, that's another consideration as part of this. So I'm kind of setting out the initial challenges there. And there are quite a few there. And that's why you'll find a lot of the precision fermentation products are a lot more expensive. And so the key here is to making sure you can put together a DSP process that's kind of robust, productive, and economical. And it's against that backdrop of those initial challenges there. I've got on the right-hand side there a few examples of the available technologies out there. And again, it is a bit of a jigsaw puzzle put together, but if you can work out exactly where your endpoint is, and then if you can bring that all the way back to the gene side, DSP becomes a lot more easier. So another thing I wanted to touch on for the downstream processing was, if you can't think about the downstream processing, if you try to isolate, say a milk protein from milk itself to isolate one single milk protein, if you look at the background to which that protein is surrounded in, there's not that much more of there are a few more of the proteins, and maybe some fats and sugars in milk, and you're trying to extract a single protein. When you think about precision fermentation, you've got all the media components, you've got the biomass, which is, doubling on a regular basis. And when you're actually these are cell batteries, so they want to survive. So they're constantly producing other biomolecules, like the strain themselves. They've got to produce lipids, they've got to have host strain proteins, they've got to have cell debris, have RNA, other proteins, a lot of other things. So what I'm trying to get out of this slide is that the extraction is very much against the background of a lot of other components. And that's really key to understand. So for the downstream processing, it's worth thinking about the physical properties of your product, but also think about also what the background of the other materials are like. So for example, if you got a number of proteases there that's affecting your protein of interest, you go back to the molecular side to see if you can have knockouts. But don't identify that after you've already done a lot of strain improvement work and fermentation. you need to really identify that really early on, so it's a lot cheaper on the development path and gets you to market a lot quicker. So I just want to put that out there. I know time's a bit conscious. So other considerations that I think are quite important, which I've kind of touched on, contaminants. So some of the products, with it being a food product, there's a defined level of nucleic acids that can be in food products. So again, having a little think about that. If there's a downstream processing step that needs to remove that. When you're scaling up products, you tend to find that there's more stress on the microorganisms, and they may produce metabolites that you didn't see at small scale. So again, that's something to think about. And depending on which host strains you pick, there will be different toxins. So that could be microtoxins for fungal-based strains. You've got exo and endotoxin from bacterial strains. And again, it's against the backdrop of lipids and carbohydrates and other proteins, which those particular strains will then start producing. My next challenge, which we've seen quite a lot on recent projects, is functionality. So when we do precision fermentation and produce egg or milk proteins, that's an easy example that people understand. You're making that protein because it's got a certain functionality. So you could use it to make a mousse or a yogurt or some kind of like meat alternative. And so you need to maintain that protein in terms of its functionality. So if it's a high gelling capability, or it can do water holding capacity well, that gives it its actually functional ingredient to be put into a food product. You don't want to be doing any downstream processing that can change the configuration. So again, that's another consideration. And that then brings me on to the regulatory and safety concerns, because when you're talking about a food-based product, you want it to be really stable over a long time, and to maximize the storage of that food material. So you got to think about, should it be a liquid product, should it be a dry product, and if you then need to take that product to market into a certain product, you may need to do some kind of like critical parameters. So that might be, say, you might need to do a heat treatment to make sure it's safe. But again, they can all affect the downstream processes and your product of interest. So I know, how long have we got, Chang? A little bit more time, yeah. So now you've considered the challenges, you're working well with your host strain team, you've worked well with your fermentation team, you've decided on your downstream processing. You then want to evaluate those downstream processing unit operations, because each one will do something slightly differently. So what I've got here is an example of cross-flow filtration. So when we design that DSP train, we then go through this process development cycle with each of the downstream processing unit operations. So for cross-flow filtration, you'd think it'd be very easy because filtration just separates based on size. But before you even start to do any experiments, you then need to look at what's available out there. And the filtration, you've got options for like configuration of your filtration rig. So it could be hollow fiber, spiral wound, or flat sheets like I've got there. There's chemistries of your membranes, and there's porosity of your membranes. So again, you've got lots of options there. We have a toolkit where we can use and pick those particular options based on our experience and based on the feedstock going into those technologies. We then go through scale down experiments that then go through 2 on the slide there. You've then got data generation, and that gives you like flux rates. It allows you to work out what kind of processing time or what kind of filter area you need. And that goes into our validation model for scale up. And then it evaluates whether, hang on, is that actually going to work at scale? So if you've got really low flux rates, it's going to take you 5 days to process. And that's really not going to be economical. Same with, filter area. The filter area is going to be like 200 meters squared of area. That's probably going to be quite expensive. So, but once you know that you've got a good answer on those outputs, you can then, as you can see there, you go to the right, then you can start scaling it through and validating those models through the scales. So that was a bit of a whistle-top story of the downstream processing. I'm just going to hand over to Chang for the final part on the scale up and the technical transfer. So over to you, Chang.
Thank you, Steve. So we've been talking about the stream whole stream selection, upstream, downstream process development. That comes very naturally to the last part of the presentation today. That's scale up the tech transfer. And hopefully by now you would all agree with us, as long as we have done all the necessary work pre-scale up, the scale up wouldn't be as difficult as one could imagine. So before we start this, I'd like to give you a brief introduction about the industrial bioprocessing service facilities that CPI owns. So we are open access facilities. We own and run a range of bioprocessing facilities that include the fermentation development lab. And also we have the analytical suite, which support the process development work and the plant scale up. We also have the downstream process development capability that we have been talking at length. We do own pilot plant facilities and demonstration facilities, which allow us to take a client's process up to 10,000 liter scales. I think the important message in this is a lot of things we talk about today. They're not just coming from paper, actually coming from the real experience of taking clients' process. A lot of the learning is through failure. So that's very useful learnings that we like to share with everybody in this room. And also, I think maybe it's worth talking about not just the facility, but it's also the knowledge and capability to do scale up, as set as apart from the rest. So on the softer side of things, we recognize the importance of the tech transfer that is reconciled between the lab and the plant. The communication is the key. We have the people talking from different backgrounds, talking about different topics that familiar with. I think today is a good example where we show the whole stream selection managers, upstream, downstream development team leads talking seamlessly about similar things. And the other thing is the external accreditations like ISO 9000 and 14000 are a huge help for us to establish a quality system, which is very important, that is actually tried and trusted. The third thing I want to mention here is that please convert as much unknown to risk. The difference between these two is that risk can be understood and so that they can be managed. How do you do this? All the activities we've been talking about in the last 45 minutes, all these de-risk activities should be happening prior to a scale-up journey. The fourth things is sometimes you do have to make the trade-off between the quality and the quantity. The sooner you make the decision, the better it is for your process development and scale up. Knowledge is power. I think Steve particularly mentioned that there's a lot of things we can learn from the farmer, from the classic process industry. There's no need to reinvent the wheel, but you do need to take into consideration of the distinctive characters of food and feed industry and apply the knowledge effectively. Last but not least, I need to stress this, as to many people before, our large-scale facility is not for testing your ideas in the process, but if we can doing a mental what-if analysis and identify as many operation risks as possible before we start the scale up, the better outcome it will be. So that comes close to the end of presentations. Maybe I just want to reiterate what is the key challenge associated with the precision fermentation on the process development and scale up. On the host strain selections, there are only a limited number of strains being approved for food use. Development of completely new whole strains is very costly and time consuming as well. And sometimes they can put off a lot of people and overthinking about the whole strain they select. But our experience is that the optimal host may not be very clear from the start. But to recognize the final product and the market it will try to crack in will hugely help to identify your back processing options. Cost, obviously, is a very important thing. We're dealing with high yield, low cost market. There are technical solutions available. The challenge is how we're going to apply this at a low cost and high yield. If we split that into two parts, upstream and downstream, they have different priorities. So for the upstream, it's your option is to match the feedstock, and you have to make very clear from the start what the preferred operation mode. Is that batch or continuous fermentations? On the downstream side, it's only about product recovery and purification. So the recovery rate is critical for most of the processes. And if you can manage to secure a product to the actual cellular environment instead of having to store it intracellularly, that's a huge benefit because that would the intracellular expression and production would warrant a cell disruption and a cleanup step that adds up to the cost. So as Steve has mentioned repeatedly, the purity is different for different kind of products and markets they're entering. So understand the purity requirement. It's very important to get the back processes right. Today, it feels like we do not have enough coverage on the formulation, but that not to say that's not our focus. We do have a separated business units at formulation. They're set up to evaluate formulation strategies and methods at high throughput and provide effective scale up options as well. So one key thing we have noticed from the preceding fermentation is that the protein, especially the protein that are produced from microbial hosts, may behave very differently to the authentic animal-derived product, and that in order to work properly, it has to be formulated carefully. So there's a lot of research that needs to be done in that regard to help identify the future formulation risk and address the risk at an early stage. Last but not least, safety and acceptance. One must know that the approval process for novel food is different across the world. So these, the earlier that you can think about your approval process, the easier it will be for your process development to scale up. And also, we have been increasingly seeing that the public acceptance of GM food is changing very dramatically. There's a lot of investment on that regard, but it doesn't seem that the past customer acceptance has improved accordingly. We see that as an important area to work, warrant for more work on that regard. So that comes to the last slide of the presentation, the conclusion and the final thoughts. I hope by going through the presentation, you all agree with that preceding fermentation process development scale up is a multidisciplinary activities, and that requires novel solution for innovations. There's a lot of knowledge available from existing technologies, and the challenge is how do you use this knowledge and apply it effectively to the new food products. And the scale-up commercialization can be accelerated by undertaking the de-risking strategies at the development stage. And we have outlined several de-risk strategies in this presentation. Hopefully you would resonate with us, which was most suitable for your own product. Particularly on the three topics we covered today, on the strain selection, the choice of microbial host need to match the feedstock process in the product. And the fermentation development work is integrated approach. It's not just a bucket and a pair of hands, a lot of clever thinking behind that. On the DSP assessment, the understanding about the final product requirement and incorporate into your development process and cycle, that's the key to your success. Okay, so that is the thank you slide to everybody in this meeting. If you have any inquiry, please get in touch. The contact details are listed here. And that's the end of the presentation. All three of us are very happy to take any questions from the audience. Thank you.
Thank you so much, Cheng, Steve, and Jonathan. That was really informational. And we have quite a few questions. So we'll try to get through with these 10 minutes we have left. We'll try to get through a bunch of these. The first one is a question about novel fungi strains. So how high or low do you rate the potential for these novel strains that have not been frequently used historically?
So I think for the novel fungal strains, I think there is really, there's a lot of opportunities there. I think in particular strains like trichoderma that have been used for really quite a long time for producing high amounts of enzymes. I think there's a great opportunity for using strains like those for producing food products. And there's a number of companies who are actually doing just that, utilizing those sort of fungal strains, using their innate sort of protein secretion capabilities to produce large amounts of protein. I think the challenges there have been to sort of engineer the strains to reduce the amount of other proteins that they're producing and eliminating any sort of proteolysis problems you might have. And you probably have some of the same issues you might have with glycosylation as well, but I think they're a really good option. Yeah.
Great, thank you. So the next questions are around feedstocks, and I'm going to group them together. There are three separate questions. One is, which feedstocks are currently used for precision fermentation and which ones will be used or possible to use in the future? I'll just let you answer that one before we go to the next one.
So do you want me to answer that one about the feedstocks, Chan?
Yeah.
Yeah. So I'd say, I'd say, well, the main aspects when considering a feedstock is really thinking about, for these processes, is often it's going to be consistency as well. So probably the most common feedstock that's currently still used is probably things like the sugar feedstocks, because those provide good consistency. You can get that product throughout the year with the same consistency to feed into your process. And the opportunities there, though, are to use sort of cheaper feedstocks. But those do come at some cost. So, I mean, in terms of achieving, like, for instance, something like molasses, that's a cheaper sort of source of sugars, and it works well as a fermentation feedstock. But you've got to consider if that has any impact on your final product. It can impact on it can come through in the downstream processing to affect sort of taste or appearance of your product. So it could, although you might make some part of the process cheaper, you might be adding some costs later on in the process as well. So I think it's looking at it in the round, and the same would go for other non-traditional feedstocks. I think they offer great opportunity in terms of achieving a more circular process, but you've really got to think about what effect it has on your whole process. And I think at this stage, in the market, it's really thinking about getting a good product out there. And I think so most companies, although they might be looking at cheaper feedstocks or more sustainable feedstocks in the future, the focus is probably on the traditional ones such as sugars. Yeah.
That leads in perfectly to the next question, which is about the sustainability aspect of producing sugar. The monocrops that are in play and the potential effect on the land. What do you see as a solution to addressing that as we move forward?
Yeah, if I could just answer this question, yeah, I think it's a good question. The feedstock, John has mentioned, currently we see a lot of process come to our door sugar-based. Some of them using molasses, which is more sustainable than just sugar itself. But I do want to stress that at this point that some of the microorganisms do not grow well on sugar. They prefer solid state fermentation or liquid fermentation, and the feedstock also has to adapt to the microorganism itself. To come back to the second question, the audience asked about the sugar. What is the environment impact on sugar? Yeah, that's a great question. I think ultimately the price of sugar will go up as the relies on unsustainable feedstock, unsustainable natural resources like land and water. We like to see more alternative feedstock to be used for alternative protein and precision fermentation. But at the moment, we don't see that many examples yet. Johnson mentioned about molasses, and I think I have seen some other presentations talking about using carbon dioxide and C1 gases as well. At CPI, we do work on those type of fermentation for food and feed applications, but at the moment, they're still not the mainstream.
Yeah, I think it's about the second generation feedstocks, isn't it? Because like you say, if you're producing crops, then that's using land that's then not sustainable for anything else. We have seen a rise in like molasses, like waste glycerol, and probably methanol as well.
Oh, yeah.
So a lot of companies are like kind of bolting onto these, building their facilities next to these waste streams to make it more economical.
Yeah. And
that's a great way forward. But again, there's probably a lot more development work because you won't get a stable feedstock. Or generally you won't get a stable feedstock coming off these waste streams. So it's good for sustainability, and it's really good to build your plant right next to these facilities. But yeah, you've got to really consider how robust your organism is to variation in these feedstocks. So I think that's really key to getting a more sustainable carbon sources.
Related to sustainability as well, we have a question asking if precision fermentation in general uses less energy than traditional fermentation.
No. I'd say in terms of the fermentation process itself, it's the same. Where it does have a less energy and low carbon footprint is comparing it to traditional sources of producing food, such as farming. I'd say that's where the comparison, favorable comparison lies. Yeah.
Okay, so we have a few more minutes before we have to wrap up, and I'm going to shift gears to someone asking about what assistance is there for startups who don't have a lot of capital on hand but are facing the challenge of scale.
So, well, for if you don't have the capital in hand, certainly for CPI, what we do offer, and I'm sure other people offer this as well, is we have a sort of bids team, and we can help companies access that sort of grant funding as well. So if you've got an innovative process, there'll be sort of grant funding agencies throughout the world that will help you develop that process. In the UK, that's primarily through sort of Innovate UK, if you want to develop a process in the UK. And we can help guide customers through the processes for giving them the best chance of winning those awards. We have a dedicated team that can do that.
Yeah, that's very good point. Thank you, John. I think maybe I could also mention that CPI also have innovation service. So that's the investment arm of CPI as well. So we do sometimes connect the client with investor as well. To those that haven't got enough capital or networks to further take up their process and product into commercialization, I would recommend, one recommendation would be is to test your process at a lab scale that is mimicking and predicting the large scale operations, and be as close as possible to the market. So try to conduct your market entry research as early as possible, so you understand what is required for fully commercialization before you're committing too much resources on a product process may not eventually work. So that's the contribution from my side.
All right, thank you so much. I can't believe how much that flew, but we still have a lot of unanswered questions. So I would encourage anyone who has asked a question that did not get addressed to please contact one of the presenters today. They would be happy to follow up with you. And that's all the time we have for today. So thank you. And as a reminder, we'll post all of this on GFI's YouTube channel, and we'll email a copy of it to all registrants. And now we can head over to Meetaway for virtual networking, and I hope to see you there. Thank you so much, panel.
Yeah, thank you.
All right, thanks.